Yes, your picture is showing.
Your screen is actually there, but I don't think it's moving. It's just stagnant.
It's just the first slide.
First slide.
Okay. Okay. Let me just stop this here. It's technology wahala.
Yeah, that's better.
Yes.
Okay. So sorry about that. I must have pressed something. So this is the chart I was referring to. So I just have to run through it again. So if you look at this is a graph, of course, a bar chart. So you have different bars. The first bar there, as you see, is the nematode bar. I think is the tallest of them all, and then it's followed. The next bar is the one for the Echitrid worms. That's the pot worms. Then you have the next chart there or bar, which is for the earthworms. Then followed by the mollusks, the slugs, and the snails. Then you have for the mariopods, the millipede, the centipede, followed by the isopod, followed by ant, followed by beetles and their larvae, followed by the dipterous and larvae as well. All these are found in the soil. By the time you do basically some soil collection, and this was a product of a research conducted across the a broad spectrum of European grassland. And what they found was interesting, and this was a big one, especially for the for the zoological sciences. That if you take just a 1,000 square, right, and if you take a square meter of soil, you would have the most abundant metazoa come up as the nematodes, because for every square meter that you take of soil sample, you are likely to have over 120 million nematodes community structure, and that is really huge. So this is just to show you an idea, just for you to appreciate the fact that though these nematodes are microscopic, but they are almost everywhere. I hope you can see my next slide.
Yes, sir.
Yes, sir.
So here we just quickly want to talk about the general characteristics of this nematode. I'll be very brief. I'll share some materials with you to help your study. These nematodes are largely aquatic, meaning, yes, you can't really find them in dry land areas as such. Of course, where there is as soon as you have any presence of water, whether it is softly aquatic or heavily aquatic, you are likely to find these nematodes there. Matthew, you are in class, so you have to. That's why I said, Michael.
Yes,
Matthew,
yes, sir.
Obaje, you are disturbing this class. Please.
Okay, sir.
We are in class, yes. Michael, unmute your microphone, please. That's the only way we are sure you are in class.
Okay.
Unmute your microphone, yes. So, as I was saying, where you find these nematodes, largely, you would always find some streams of aquatic life, if you want to put it that way. These nematodes are unsegmented. By unsegmented, imagine the earthworm. If you look at an earthworm, for example, the first thing that draws your attention are the segments on the body of the worm, clearly particularized segmentally with all those line. Unlike the earthworms, nematodes are not segmented. They are unsegmented. Matthew, please control the noise from your end. Nematodes are unsegmented. They are smooth. They are transparent. You could see all their organs by just looking at them under the microscope. You could see their reproductive system. You could see their digestive system. I like to say it. Just imagine I'm looking at MJ, for example, or I am looking at Mary, and I can see Mary's ovary. I can see her intestine just by looking at her. That is the level of transparency you find with this nematode. They are transparent, meaning you can see all their organs, all their internal organs. So unlike most organisms that you don't get to find that with. Of course, there are other metazoans too that possess this attribute, but largely nematodes are, and they are unsegmented. They don't have appendages like leg, all right? They don't have those external appendages as such. Then they are bilaterally symmetrical, meaning if you split them into two, you will still not be able to, it's like almost a replication of their body. For example, think of human being. A human is bilaterally symmetrical, meaning that if you split us into two, you will still find some parts appearing on both sides. For example, the eye. If you split man into two, you can still find eye on one side, find eye on the other side. You can still find nose on one side and find nose on the other side. You can still find almost everything you find. There are some organisms that are not, symmetrical. So, or bilaterally symmetrical, meaning if you split them, you will find a different organism. Matthew, you are really disturbing this class, Matthew. Can you be stable? Please, if you are joining an online class, the first thing to do is to locate yourself where there are no noises. Otherwise, you are going to be disrupting the class in a very annoying way. Please, Matthew Obaje, maybe
Yes,
you need to control the noise from your end. It's too much, please. If you can be stable.
sir.
This class is only for 40 minutes, so please, let's just keep everything.
I have the lecture. Thank you.
Okay, very well then. They are generally bisexual, meaning that, well, for most nematodes, they don't need spouses. So if the spouse is there, they can reproduce. If they don't have the opposite sex as well, they can still reproduce. We refer to such phenomenon as biosexuality. Then you have the vermiform. They are vermiform in nature, meaning there's like the picture you see here. You can see them looking so long. They look like small sticks, just round sticks. We call that vermiformity. They are vermiform, but there are others, depending on their life cycle, that are not appearing this way. You will not even know that these are even nematodes. They can assume the shape of a kidney, the shape of an apple, depending on the life stage that they are at, right? Then there are non-coelomates, meaning within their internal structure, they lack the coelomates. Then the biotroph, please, just a minute.
There's pleroma there. Hello.
All right, thank you. Sorry, I had to respond to a call from the reception. Okay, so lastly, they are biotrophs, meaning they feed on biological materials. So that biological material could be plant material, it could be other biological material in the environment, like it could be that they are feeding from a collection of bacteria, or they are feeding from a collection of fungi, or they are feeding from a collection of other nematodes, because you have a group that are predaceous. I'm going to take you through very quickly. You will see some pictures so that we will try to have a full concept of these things. Now, these nematodes, as we know it, they have a very broad environmental spectrum. Meaning there are those that are free living, those that they are just existing in the soil. Basically, they are decomposing materials. You have those that are not free living. By free living, of course, what we are saying is they have no business whatsoever with plants in the environment. They have no business whatsoever with anything at all. So their own focus is just for their own existence. They contribute to the ecological components of the environment, meaning they can be involved in the degradation of some materials in the environment. There are those that even contribute to the oxygen content of the soil environment by their movement in the soil. They can torrent, meaning they can create room for soil aeration and all that. So we refer to these free living ones as just being free. They're just doing their thing. But there are others that are doing their thing, but it's affecting man. Those ones are referred to as the plant parasitic or herbivores, if you want to put it that way, because they feed on plants. I will take you through some of those ones very shortly. So how do these nematodes respond in terms of their sensory system? You know, I've mentioned earlier on that they have a very simple body system, just digestive system. They have a reproductive system. They have an excretory system. But in terms of how they're able to sense gradients in the environment, for example, if there is heat, we would all say, oh no, this environment is hot today. Or if there is cold, we know that there is cold. So how do these nematodes also know or have clues in the environment when things have changed? They have what is called the arm feed. You can see from this picture. I hope you are able to see this, my slide. Can you see, please?
Yes, sir.
Yes, sir.
Yes, sir.
You will see very clearly this thing that looks like a ring on the head of the nematode. By the way, this is the head of a nematode. This is even a marine nematode. So you will see very clearly something that looks like a ring on the head there. That is called the arm feed. With this organ, they are able to sense if there is any chemical activity within their environment, if there's any change in osmotic pressure. And then once they're able to do this, of course, information they say is power. Then based on the clues that they are getting, they can take certain decisions to either avoid or run, as the case may be, or stay if it's favorable. I just wanted to start showing you some of these things so that when we get to other parts, it will be easier to explain a few things. This nematode, by the way, is called the Achromadora species. Very beautiful nematode, if you ask me. Very beautiful.
Is it a free living type, sir?
Yes, but this one is in a marine environment.
Okay.
So you find these ones from the sediment of a water body. So you know that every water body has a sediment. For example, River Benue. If you get to the bottom, to the last bottom, there are soils there,
Yes.
right? That is known as the riverbed. It's known as a riverbed. So once you collect samples from there, you find this kind of nematodes there. This is this is an elaborate, so to say, this is an elaborate picture of this nematode. For example, you find there are two of them here. You see the first one. This is usually the male. The males are smaller in stature compared to the females, generally.
Yeah.
So you find the female here, and you can see all the body parts. I'm going to share this with you. I'm not sure it's that clear, but no big deal. It's part of your assignment today to show a diagram of a plant parasitic nematode showing all the component parts of the nematode. But so this is intentional. I'm just showing you some things that I expect you to also provide during the submission of your assignments, especially for MJ and Matthew. So, but of course, Michael and Mary, it's fine if you also want to do this. It will help your understanding of this nematode anatomy, really. So don't forget, nematodes lack circulatory system. They also lack respiratory system. And then the big question is, if they lack circulatory system, if they lack respiratory system, how then do they move things within their body? How then do they also take in and give out exchanges in terms of gases? So one of the exam questions that comes is, nematodes lack circulatory and respiratory system. Explain. If you see that kind of question, what it means is, yes, you would also tell us, then how do they carry out circulatory and respiratory activities. If they lack these systems, what are the structures in place that ensures that things, especially liquids, either in form of lymph or blood or whatever, is circulated within the system of the within the nematode body? How are they able to breathe? How are they able to take in and give out oxygen? Right, so this is where we are. Don't forget, this is a master's class. At master's level, understand that you are not going to be taught everything. No? So to a great extent, you have to do a lot of study. And I've packaged this assignment so that you can also get to that point. So please, the question here is, nematodes lack circulatory and respiratory system. Explain to me, how are they able to, how are they able to circulate things within their body? How are they also able to breathe or carry out any exchange, any gas exchange within their body? That is the second one. The first one, as I said, is draw a schematic diagram of a nematode showing all the component parts. Please, not some. At undergraduate level, you must have been shown one that has maybe some 12 parts or whatever. You can see all the parts here in this diagram, and it's intentional. They don't come out very clear. Even if you print, you won't see the name like that. So I expect that during your own research, you're able to come up and know exactly what those parts are. But I must tell you this, if you look at this nematode, you see the head region, you also see the tail region. The head region is very simple. Whenever you see anything that looks like a stylet or anything that looks like a mouth part, because it's difficult to tell from a microscope if you are not an expert, to tell where the head is from the tail. So if you look at this, can you see my arrow? I don't know.
Yes.
Yes, sir.
Yes, sir.
Yes, sir.
If you look at the area that my arrow is revolving around, you can see from the head region, you can see that there are some scleroterized kind of structures there, which tells you that that is the stylet. And this is known as the anterior part of the nematode. This tail region is known as the posterior part of the nematode. And at the posterior part of the nematode is where you usually have the anus, or what we call the rectum. And for the male, it has what is called the spicula. These two things you are seeing here is called the spicula. It's like the penis in a man. Then for the female, of course, you also have the rectum. Basically, you also have some other part. Let me not even start spelling things out for you. So, but please, I think the most important thing is to understand the anterior part of the nematode, and that it also has a posterior part of the nematode. And why is this important? With the anterior part of the nematode, we are able to identify the nematodes to the species level. We are also able to identify the nematodes to the to the general level, meaning, you have the genus, you have the species, and on in the taxonomical class. With the tail as well, we're able to identify nematodes. So we have over 12,000 species or thereabout for plant parasitic nematode, and over how many hundreds of thousands of species that have been described. How are we able to know which is which? It is by describing some of these things. In fact, some are so detailed, they will tell you that the length of the stylet alone can be used to tell a particular species of nematode. For example, the nematode I'm showing you here is certainly a plant parasitic nematode, and this look pretty much to me like even Melodogyne. And then you find a situation where a particular species, Melodogyne has over how many hundreds of species already described. There's Melodogyne javanica, there's Melodogyne hapla, there's Melodogyne hanareria, there's Melodogyne enterolobi, there's Melodogyne incognita, there's Melodogyne hippia, a whole lot of species of one genus. It is in understanding the parts and the lengths or the positioning of these parts that they are able to describe these species. But that is not the focus of this course. The focus of this course really is to begin to look at these nematodes with respect to their environment. Don't forget, this course focuses on ecology of nematode and their control. But control is the second component. This first component is on ecology, largely. And I showed you here, for example, Pratylecus. There's the first one I'm showing you under this first plate. You see, this is Pratylecus. The first picture here shows you the entire nematode, which is A. The second picture shows you the posterior part of the nematode. The third picture shows you the tail or the posterior part of the nematode. Right. Then the last picture here shows you, okay, oh, this looks to me like a male. It shows you the male. Don't forget, I told you that the males are smaller in stature compared to the females. So that A there certainly will be referring to a female nematode. There are some things I can see here that you may not be able to see, but that one is largely due to experience, because I can tell if I look at this region now, I can see something that looks like a vulva. There is one black spot here that looks like a slit. It's a very short, dark spot. That tells me there is something happening there. It looks to me like an opening of some sort. Now, for you, it may just look like everything is black, but there are some black that are strategically located. And I also know that the vulva is around the 50 or 70% of the entire length of the female nematode. So that is where you find the vulva. The vulva is like the vagina of a woman. So that is where you find the vulva opening. And if you go down there, certainly you find the rectum or the anus. And for the male too, you see, the spicula or the spicule is around this region. So what I'm saying is, if you want to identify a nematode, you don't just look at the nematode and say, yes, this nematode is this. This nematode is Pratylecus Z. This nematode is Pratylecus brachyurus. No, you must get certain parts in place. For example, you want to look at the entire, are there females? Look at the female, look at the male. And you take the anterior parts, take their posterior parts, and begin to look at the organs that are there. How do they fit into a Bible, what we call a description or a taxonomical Bible, for example, if you want to put it that way. So, or we call it a nematode identification key. It's like a book that has all these nematodes that have been described. So as you are looking at the nematode in the microscope, if you suspect that this nematode is likely Pratylecus, you open that part of Pratylecus on the book and start comparing. And the one that falls closest to the description you envisage from the microscope is likely the genus. All of you here, except maybe Matthew, you've had cause to send your samples for analysis and all that to see, okay, what are the nematodes in this my sample? These are the things they do. They get a nematode identification key. They also get very sophisticated microscopes in their labs and gadgets that can help them take clear pictures and also measure parts. It takes a lot of work. It's not that easy. Maybe if we have an opportunity to visit one of these advanced labs in the future, we should be able to organize and do that. But of course, the economic situation and all that is not making things funny. And making such moves is becoming a challenge. But we can get around it by some form of technology. I will also see how I can share some links with you where you will see real, they call it virtual lab. So even from YouTube, you'll be able to zoom in into a nematode pattern, zoom out and have a feel. So the next nematode there is Zephyrema. Zephyrema has a lot of species that have also been described. Then you have the Criconemella as well. This one is one of the most beautiful nematodes I know. It looks like an earthworm. And somebody will say, but okay, you said this thing is not segmented. Is this not some form of segmentation? No. What you see here is an adaptation. It is a form of adaptation that this nematode has built. It has its ability to zigzag into these kind of structures you see that makes it look so thick. So they are not easily parasitized like the others. Don't forget, this nematode in the soil too, just like we run away from lion, they are also escaping from some particular nematodes. Talking about how versatile they are physiologically. Yes, nematodes are versatile. They are able to tolerate harsh habitats. Harsh. Michael Lupa is working on isolating some nematodes from polluted areas in Makurdi metropolis, for example. And then one will wonder, this kind of harsh place, which organism will be living there? I tell you that once that thing is out, you'll be shocked at the kind of nematodes you'll be finding there. Mary Dauphin too is also working around that sort of vicinity, but she's focusing on pollution sites in abattoirs to see what kind of nematodes can we find there and can they be used to monitor pollution in the environment. Those are the questions. So these nematodes have the ability to go through some of these harsh environments that some other organisms will not be able to tolerate. And that is one of the reasons it's been said that nematodes are the highest when it comes to metazoan on Earth. They are the highest metazoans existing on Earth because they can tolerate quite a range of habitats, even down to the Sahara Desert. Just have a small drop of water and you find them there. And it's also interesting to know that, yes, of course, there are interspecific competition, yeah, for most of these other organisms. You can have that too for nematodes, but they try as much as possible to avoid interspecific competition. So in just one sample of soil, you will find all manner of nematodes there. I want you to think of this as coming to Makurdi, for example, and seeing Japanese, seeing Australian, seeing American, seeing a red Indian, so we are all just gathered there. Nobody is kidnapping anybody. Right, so that sort of mix is what I'm talking about when we talk about nematode community. It means a community where you find all manner of nematodes there, just surviving. So they try to avoid interspecific competition. And of course, many environmental selection pressure as well, meaning that if the environment goes harsh, they have strategy for survival. I think I will walk you through that one too. And they have the ability to regulate the uptake of oxygen. Imagine. So whether oxygen is available in the environment up to 100%, they can survive. If the oxygen availability is about 5%, what it should be, they can also survive. That's a very wide range. And I can tell you that most of us cannot survive oxygen environment that is less than 70. Some persons cannot even go beyond 50. Not to talk about 5%. Right. So this is one of the reasons why you are in a room, and let's say there are many people in that room and you stay in the room for late, let's say 1 or 2 hours. Some persons will start running outside because they start feeling very funny. And at that point, the oxygen availability is not even up to 50. So, but for this nematode, they have the ability to uptake oxygen between 0 to 5%, and then they can still go through their normal physiological activities. They are very permeable. They have a hydrostatic skeleton, which helps them to, regulate calcium, magnesium, potassium in the environment. And very interesting also, they can tolerate a wide range of pH. pH ranges from 1 to 14, isn't it? 1 being acidic and 17 being, what am I saying? 7 being neutral and 14 being alkaline. Now, this nematode can go between 1.6 to 11, meaning they can survive that environment. So this also explains the reason for their survival and their spread in nature. Then you have those that have the ability to also, survive temperature from sub-zero to as high as 60 degrees Celsius, and some even more. This reminds me of the thermophiles. And it's crazy because what this simply means is that you cannot even use temperature to control some nematodes. And that has implications for management. Because if some of them are easily killed by temperature, you can say, okay, I will use temperature to destroy them. But depending on the nematode concern, Some have the ability to even stay on in a wide range of 60 degrees Celsius plus. And if you say, okay, let's reduce the temperature to the point where they freeze, they also have the ability to survive freezing temperatures. So, because one of the ways we also keep things or preserve things is by freezing them, isn't it? We freeze them. So if you want to use that, there are nematodes that are also able to withstand that much pressure. And we call that sub-zero temperature, meaning going below -1, -2, -3, -4. Yeah, they can cryolite. So I wanted to show you pictorially one of their survival strategies. So what this nematode here is called Aphelenchus. And Aphelenchus has the ability to carry out what is known as an hydrobiosis. And hydrobiosis is the ability of an organism to lose almost all the water it has in its system and enter into a state of reversible metabolism. I'll take it again. Anhydrobiosis is the ability of an organism to lose not all its water, almost all its water, right? And enter into a state, right, of reversible metabolism. What that means is metabolism is going on, but not in the normal way it should go. So with this potential or ability, they're able to stay for longer periods of harsh environmental weather. So when the environment becomes okay, they bounce back and then continue problem.
Just like other species of animals hibernate.
Yes, it's a form of hibernation, if you want to put it that way. But we call it a for hydrobiosis, this one refers to is water. For hibernation, it could be that they just reduce an activity. For example, if they were moving, they would decide not to move again. So they would just stay in one spot. But for anhydrobiosis, it is about the water content. Because don't forget, I started off by telling you that these nematodes are found wherever there are aquatic, any form of water or wetness. You find them there. So water wetness is a big deal for them. So if a nematode has the ability of anhydrobiosis, I mean, that means it can survive over a very long period of time. I showed you here the hair structure of different soil nematodes. And of course, this gets interesting. So in the soil, you have the bacterivore, you have the fungivore, you have the omnivore, you also have the herbivore, and then you have the predator. And then the question here is, what are the classes of soil nematode based on their feeding habits? So based on their feeding habit, this nematode can be categorized into 5, right? So they can be categorized as bacterivore, meaning that these are the ones that feed on bacteria. For example, look at this diagram here, A, and you can see how their head is. A, for example.
Yeah.
B, for example, that has something like a crown on its anterior parts. Even C, but C has double ability. Because if you look at this black thing inside here, this is the head of a nematode, don't forget. But this is a different genus. They are all different genuses of nematode. But I don't want to be boring you with their names now. But if you look at this black thing inside, this is like the tooth. This is like your teeth. Some nematodes have teeth. They don't have stylets. Stylet is, look at D. Stylet is this one right here that looks like an arrow, sharp. Some nematodes don't have this. Some nematodes don't even have any structure inside, like A. Look at A. A doesn't have anything, just normal opening. So A can take in bacteria, swallow in bacterial cells, and digest it. B also can do the same, but for B has some mandibles, some structures just aligned the wall, or rather the point where they are taking things in. But for C, you can see that there's a presence of some like tooth-like structures there. And then D has like a stylet. In fact, if I ask you from A to B, which of them is plant parasitic?
D,
Why D?
because it has stylets, sir.
Very well. Is this who is talking? Is it Matthew?
Yes, sir.
Okay, yes. Yeah, so you are correct, Matthew. The presence of a stylet signifies that that is plant parasitic indeed. But don't forget, there are different types of stylets. If you come down, I'll show you a different type. I'll also show you in details very soon. So this is just to note that all these categories are basically bacterivores. D is not bacterivore. D can either be a fungivore, it can feed on fungi, but it can feed on plants. So anyone goes. E is an omnivore. You can see it has the presence of a very thick teeth here, looking very sharp. And then it can feed on anything. It can feed on plants. It can feed on other nematodes as well. It is omnivore. It can feed on anything. Look at F. F is an herbivore. This long thing you are seeing here is a stylet, but it's not looking like an arrow. It's looking like one long thing from nowhere. Right. In fact, the first time I saw this stylet, I was like, my God, which kind of stylet is this? It's so long. And yes, it is so long because this particular nematode here, in fact, let me give you the name. This is Zephyrema. This is Zephyrema index. You have Zephyrema, they have this sort of very long extended stylet. You have another group known as Longivorus. Longidorus, they have this very long stylet as well. Trichodorus, they have very long stylets. And because these are the group of nematodes that parasitize on trees, those trees, I mean, if you look at a normal tree, the roots are very, very hard. So they have also been able to adapt their stylet to be able to penetrate that root. So as thick as that root, these nematodes are able to penetrate with their thick stylets that is so long so it can get right in, and then they can begin to suck the content of the plant. So that's a herbivore. Then this one on G is also a herbivore, beautifully tacted, and then H is a predator as well. So for a predator, you can see how particularized the teeth is. Really thick. In fact, it's the most advanced of all of them. Because for a predator, you have to be shown to have the capacity to eat anything. Their head big, everything big. Just think of a lion and the teeth of a lion. So look at this black thing. It tells you that this one can grab any other fellow nematode and take it all in. It is squarely feeding on nematode, nothing else. It doesn't feed on any plants. It doesn't feed on any bacteria. It has no garden time for any fungus. It's going for a fellow nematode. I want to show you here a typical example here of a predatory nematode. So this is a real nematode. This is a real nematode picture. This is taken from the head region, and this is from Peter Pauling. This is not my picture at all. This is from Peter Pauling, and this is the head region of Mylocolus montanus. This is a predatory nematode, and it's showing you magnification up to 1,000 times. This is a 1,000 times magnification. Again, this is Mylocolus montanus. And you see how thick the tooth is, very thick. So once it grabs the nematode, it budges it and one time dies and flushes it into its system. So they will feed on nematode, as I said here. Now, there was this study that showed from Peter also that, yes, these things feed on nematode, but when they don't have these nematodes available, they also have some strategy in place to be able to feed on other protozoa or bacteria. I told you in the previous slide that they were first described this way. But this study in 2000 came to show that as against the previous knowledge that these predators squarely feed on other nematodes, they have also structures in place that show that they can feed on protozoa and other bacteria. So this is a case of when the available is not available, the available becomes when the desirable is not available, then the available becomes the desirable, isn't it?
Yes, sir.
Then you have those ones with the simple tubular mouth parts. These ones, they feed basically on bacteria. All right. So they just take in the bacteria and then they are able to digest. They have a very elaborate cuticle around the oral opening. So they're able to gather as much bacterial cells as possible. This is also from Peter Mullin as well. What about the plant parasitic ones? As I said earlier on, you can always have, they can come in form of arrow. Look at this first one. This is the one that comes very sharp, stylet, exactly. And you can see the stylet knobs here. And then once they are able to penetrate, they push this stylet, just think of, you're pushing it out, penetrating. And once they're able to make their way into the plant cell, they begin to suck the sap out of the plant. And of course, the plant begins to wither. This is the second one with a very long stylet here. Very long, really long. You see, it doesn't even have a stylet knob like this first one. So it can go distances. It can penetrate distances and then begins to feed on whatever it is it needs to feed in. The life cycle of a plant parasitic nematode is just about 21 days. Not long. That's about 3 weeks, isn't it? So they can do like 3 weeks up to 2 years for some nematodes. For example, the dagger nematode. The dagger nematode can do like 2 years. That's crazy. So, but most of them within the range of just 1 month, they are done with their life cycle, like Melodogyne. But those ones that have very short life cycle, they are even the most problematic. They are our problem because they want to produce everything, kill everything before they go. So again, what they do with their stylet is just probe. They probe for what to eat. But in the process of probing, they destroy these plant roots, and the plant begins to show symptoms, and then it's all gone. Then these nematodes can be ectoparasitic, meaning they can be outside and complete their life cycle, or they can even complete their life cycle within the plant itself. I'm not going to bore you with all these. These are just generalistic presentations here. But just to show you here that host range can be from 1 to 100. That means it's not as if you say, okay, for only tomato, it's only one nematode that affects it. It can be sorry, not to say Melodogyne, for example, only affects one plant. No, it can affect a number of plants. One nematode alone can cause a number of problems in other plants. Just one. So you have those things playing out from time to time, and just basically it anyways. And to note here that all crop plants are susceptible to at least one nematode species. Of course, most are root parasite, mainly they carry out their parasitic activities on the roots, and then more damage can be associated with soils that are coarser in nature, soils that have a lot of large pore space, because once there is a large pore space, the nematode is able to thrive, is able to get more oxygen and cause more problem. So like clay soil, for example, that has almost zero pore space, clay soil is difficult to have some of these plant parasitic issues playing out. But the problem is even the plants too don't like clay soil. Yeah. So, but then you have other group of nematodes, the free living ones that love clay soil because there nobody is disturbing them much, and they're able to see their other friends to eat from. So what are the major impacts of this nematode? As I earlier mentioned, they are involved in the decomposition of organic matter, and of course, the recycling of these nutrients in the soil food web, right? They are also can serve as biological agents, right? Yeah, especially for insects. There is a group of these nematodes called the entomopathogenic nematodes. They feed on insect larvae. So if they are able to feed on the insect larvae, that means the insect is not able to metamorphose into full adults. That way you would have controlled it. Right. Research also has been conducted using this nematode. For example, Sinorhabditis elegans is the first nematode that was, in fact, is the first organism that was used to synthesize or sequence the DNA code of human being. That research was a breakthrough research that changed the landscape of molecular biology completely. You can browse it. C. elegans. is a nematode that was used to study biological model. For example, what does this mean? If I want to know the genes that are responsible for sight, now you have technology in place that can knock out that genes and it can make the person blind genetically. I'm not saying physically, because if I say make somebody blind, you then puncture the person's eye and the person goes, that is not a genetic thing, it's a physical thing. It's an environmental anomaly. But a situation where you do it such that if the person gives birth, the person's child or progeny will also be blind. So that particular study was very interesting because it made scientists to understand how the genome of a human really is. And they were able to sequence all the genome of a nematode. And it was like, if we're able to do it in this organism, that means we can do it in humans. And as soon as they were able to do it for humans, the thing took on another turn. Now we have labs in China that have kidneys for cell. They have all manner of organs for cell. And I don't know if those ethical issues have been dealt with, but it's crazy now what we have. What about this impact again? They can also be used to study disease of animals and humans. For example, the heartworm. What about trichinosis, hookworm, etc.? These are all nematodes playing out in humans. And in fact, there's a disease called elephantiasis, which you must also know about. And that's a nematode problem, by the way. So the nematode goes right into the vein and begins to cause a lymph node there. So that way, nothing is able to flow and the legs begin to swell.
Yeah.
I think I've seen 1 or 2 persons with that, and it's really not a good one. Yes, there are also important plant pathogens, no doubt. For the animal parasites, you have them, because as I said, they can result in night blindness, right? Elephantiasis, and it's crazy. In our pets, hookworm, in our insect, they can be used as control. See this picture here. You will see some things inside the insect. Those are not parts of insect, those are the nematodes insect. So this whole thing is an insect, but the nematode is able to gain entrance either through any of the openings. It could be the spiracle of the insect, it could be the anus of the insect, whatever it is. Once they're able to gain entry there, they begin to multiply, and then they begin to feed on the internal content of the insect, and the insect dies. So all those things, whitish things you see inside, those are nematodes. And we refer to those groups of nematodes as entomopathogenic nematode. So this is the Sinorhabditis elegans I told you, the second picture you see down here. This is the Sinorhabditis elegans I told you. And this is the first nematode that was used as a basis for studying different genomic whatever. For example, this was used in studying aging. In fact, the professor that led this research, I really wanted to work with him, but my brain was not hot, so he didn't select me. I was really, really interested in his line of research. He worked on aging. And I think I should go and check. In fact, now that I'm teaching this, I should go and check him up. Let me see if he has aged. Because he was so passionate about this. And then the whole study back then in 2012 was, he was coming up with this strong solution that would fight against aging. And then, I don't know, but it would be good to check him out and to see what's happening there. Why some persons are saying that aging comes with, aging is not the same thing as growing old. What am I saying? Aging is not the same thing as adding years to your years, right? We're looking at aging in this context as getting old, the tissue becoming weaker, right? And then studies there were focused on how to use this Sinorhabditis elegans to fight aging. If it can slow down the aging process of Sinorhabditis elegans, that should also work for humans as well. So I really wanted to be part of that research group, but my GPA was not good enough for him to take me in. I think it took like 2 persons, but I should check them out if all of them are not old now. Neurology, ecotoxicology. These are areas, very interesting areas that Caenorhabditis have been used to cover. For example, what is the effect of a chemical on a biological organism? We'll talk about that in the light of LD50, the lethal dose 50. To what extent can damage be caused? You can't try that with human beings, right? But with this nematode in place, you should be able to, just do one or two things, yeah? Yeah, you can carry out these studies with Caenorhabditis elegans. Okay, so I think the last slide here will just be to show you some basic nematodes that this is just a schematic presentation, right? You will see that nematodes occur in different shapes and sizes. For example, look at number 1. This is number 1. Very long. This is long. In fact, the name is Longidorus. It's very easy to remember. Very long. This nematode is so long and it's called Longidorus. We're just looking at the size, the shape now, nothing more. No any details I'm showing you here. Look at number 2 here. Now this is Dolichodorus. Dolichodorus is also a long nematode. Then number 3, you have Belolimus. You have Anguina. Number 5, very interesting. I had to highlight this in a slide. This is Zephyrema. Zephyrema is interesting. Those are the nematodes I'm really focused on personally now in my own research. Then you have Hoplolimus is also there, number 6. On number 7, you have Rotilencus. On number 8, you have Hemicyclophora. On number 9, you have Dictylencus. On number 10, Mary, what is number 10?
Hey, God.
Hey, God.
Okay, I'm trying to. Okay, is Dictylencus.
Is what?
Okay,
Allow Mary to talk.
Aphilencoides, sir.
Very good. So it's Aphilencoides. Michael, what is number 11?
I see I can't see very clearly. But...
No, you can see it clearly. You can see clearly, I'm sure.
Tylencohicus.
Tylencohicus.
Yes, so it's important we have all this because we need to be used to the names, and this is good because you are all researchers now. MSc is a research degree, isn't it? So yes, you're right. That is Tylencorhynchus. Number 12 is Trichodorus. Number 13 is Rhadophilus. Number 14 is Pratylecus. Number 15 is what? Matthew, what is 15?
Pratylencus.
15.
15 is Criconemoides.
That's correct. Criconemoides. Yes. That's very correct. Number 16 is Pratylecus. MJ, what is 22? Sir? Number 22 is what?
Helicotylencus.
Helicotylencus. That's very correct.
Yes, sir.
Who else has not spoken here? Who is on this?
Everybody. Pleroma is out. Her network is so bad.
Oh, too bad. All right, that's fine. So, MJ, what of 21?
21 is Rotilencus.
No?
Okay, Rotilenculus.
Rotilenculus. That's very correct. Number 20 is Criconema, and number 19 is Tylencus. Then you see Melodogine. This Melodogine you see here on number 18 and number 17, Heterodera. Now, why am I showing you these ones? You can see that, remember when I started this class, I told you that nematodes are vermiform, meaning that you just think that they are looking like earthworm-like or worm-like. It's not always the case. Look at number 22, Helicotylencus, for example. Number 21, Rotilenculus, for example. That's not a vermiform structure. That looks to me like a kidney, sort of. Right.
Yes.
Look at number 19, Tylenculus. Tylenculus doesn't look to me like a worm. It looks to me like a a lung of some sort. Look at Melodogine and Heterodera, 18 and 17. They look like apple to me, orange-like, you know. In fact, the word Melodogine is gotten from the description of an apple.
Okay.
So, yes, they are vermiform, but at certain stages of their life, they can take on different forms, or by default, they are that form. Melodogine doesn't start off this way. It is the female that you see there, even Heterodera. That is why I put them in the box. The both of them is the female, and it shows that they are pregnant. Once you see a structure like this, it shows this is a pregnant Melodogine or Heterodera, depending on which one now. Right. And is this not the same thing with humans? If a woman is pregnant, she's not the same form anymore. Right. So we see some of these things playing out in nature that tells us that there is more to life than we see. So, ladies and gentlemen, we have extended the stay of, sorry?
Is Tylenculus pregnant too?
No. Teleculus is not pregnant. That is the shape. That's why I said it's only these ones in box that are females. They are pregnant females.
Okay.
When they are not pregnant, they assume the worm-like form. But when they are pregnant, you see them looking like this. Of course, like a pregnant woman.
Now, what's the reproductive rate averagely? Like you said, they exist in millions. Or
Whoa! Talking about reproductive rates, like Melodogine in a single, when it's pregnant, it can go as high as 20,000 eggs produced.
Wow.
So how do they get pregnant?
How do people get pregnant in real life now?
Either sexually or asexually, as you said.
Yes, look at this. In fact, yes, so they can, you remember we said that they are bisexual in nature. So when the male is available, they copulate. They also copulate. I will share some videos with you. You will see where, in fact, you can even look for those videos yourself on YouTube. Copulation in nematode. You will see how they make love too. Right. So they carry out the normal reproductive activity that results in this. Just normal. And then when they are producing, they produce so much, over 20,000 eggs, because they are not sure which one will survive. So they produce so much, and then not all of them survive anyways, as you expect, but they become a nuisance in the environment for those that are plant parasitic nematodes. For those that are free living, we find that they are highly beneficial to the environment. And in fact, we are increasingly trying to find ways of how we can take advantage of them and help so that they can help us understand our environment better. One of the things you must have taken from today's class is that these nematodes occur in a wide range of ecology. Not all of them are disastrous. So when you hear of nematode, your mind should not just go to problem. Yeah, it depends on the nematode involved here. Some nematodes are beneficial, especially the ones that are free living in nature, while others are parasitic, those ones that feed on plants. Others are even animal parasitic, those ones that cause harm in humans, right? So these are some of the different types of plant parasitic nematodes. As I earlier mentioned, some are ectoparasites. That means they feed from outside the plants. But they do this in two ways. They can move, right? They can migrate, yes. Some don't move. Once they enter the plant, they stay there. They are sedentary. So they can just enter and do what they need to do. Then you have the endoparasitic ones. These ones, they also have the migratory one. You have the sedentary ones. For the migratory ones, all their body, everything is inside the plants. They keep moving inside the plants. For example, Pratylecus, that's another area of my focus, legion nematode. They keep moving within the plants. As they move, they keep destroying tissues. While you have those that are, once they enter the plants, they will just settle in one spot, like the root knot nematode and all that. So just to show you a picture of them, you can see the sedentary ectoparasitic ones. So look at the arrow part there. Here, you can see that this one is ectoparasitic. The head is outside, but you see the stylets penetrating into the plants. Look at the stylet here, this long thing here, penetrating into the plant, and they begin to suck, but they don't enter. But look at the sedentary endoparasitic ones. All this red thing you see, all these things in red is a stain. So that's one way of finding out if the nematodes are there. That's what we call the red stain, the blood, you call it the blue stain. You can stain a tissue. Once anything stays, it means that there is a living thing inside. So if you look at this stain, you see very clearly. Who can tell me this nematode that is here? Look at the shape now.
Like, I can't remember the name. The one that was just outside the box. Those two that you boxed.
The one just before the two in the box.
No, so the ones inside the box, right? That's look at them now. Look at it.
Okay.
So it can be any of
Melodogyne.
so it's either Melodogyne or Heterodera. We don't know, right? But from here you can see they say it's Melodogyne.
That means like sugar cane. The host looks like
how does it look like sugar cane? How? But look at the structure here. This looks to me like what you find here is the egg. Now, this is a pregnant woman that's giving birth. All these things here now, these are the egg. It's called the gelatinous matrix. As I said, this one is just based on experience. But this is just to show you that they are right inside the roots and then they are sedentary, just feeding from a spot. But look at this last one, the migratory endoparasitic ones. They are inside the root. Look at them, red. They're just there moving, eating, destroying as they move around. They don't stay one place. Right. So this is the last slide now, just to show you a typical life cycle of a nematode. So it begins from an egg. So the egg is laid, and then it moves to the J1. The J1 is juvenile. Usually we call it juvenile in nematology. We don't use the word larva. We use juvenile. J1, J2, J3, J4, then adults. Right. So it goes through this molting stage, just like an insect. Nematode is related to insect too. It molts. It also molts. So from the egg, it molts. That's what this M refers to. It molts into J1. Yes, into J1. Right?
Yes,
Then you have the issue of J2. Then J3.
So what does H mean in front of
J4? Hatch.
Okay,
hatch. Okay. I think it's there now. Look at it. It's up here.
The keys are there.
Now, this H is not referring to this arrow. It's just showing you what is happening here. That this is where the whole thing starts from now, isn't it?
Yes.
So usually when you have the egg, the nematode has to come out. That process of the nematode coming out is called the hatching process. So the nematode comes out of the egg. And that's hashing. When it comes out, the first thing it assumes is the J1. It is the J1 that hashes from the egg. Then the J1 moves on to J2. The J2 moves to J3. J3 moves to J4. And then have an adult. This picture that is here shows you a typical, shows you a typical molting process. You see, this is the old skin. This is the new skin. This is the new. So this first one here is old. It's coming out of the nematode. The skin is leaving the nematode. And what you find here is a new one. So all these things will go, including the stylets. They will all fall out. This is in a schematic diagram. Now, this is the typical life cycle of a Melodogyne hapla. Right. So as I said, the whole thing begins from the formation of, or the egg, basically. Right. So it goes from the egg, which is down here. As I said, if you remember this picture here, that as I said, that this is the female and this is the egg. So the egg is released. From the egg, it moves on to the J1. This is what the J1 looks like. It's still in the egg, but it's trying to hatch. And then by the J2, this is the J2 here. This is typically how the J2 looks like. And it is the J2 that is a problematic stage. This is a stage that starts looking for a host. Because the egg are laid outside the plant. They are not laid inside the plant. They are, you can see the nematode. This is the roots. This part here is roots. But this thing there is outside. So the egg is laid outside the plant. So J1 is outside the plant. It is when it goes to J2, it is the J2 that begins to look for a root to penetrate inside. And once it does, the J2 penetrates into the root, see them inside, all scattered inside the roots. And then they look for where to feed. Once they get where to feed, they form what is known as the giant cells, or what you now find as gods. But don't bother yourself with this. It's not really important now. It's just to walk you through the different stages. Then you have the J3, which is really, really big now. And then you have the J4, which is more or less like getting to an adult. And then you have a full adult stage. The adult again, this is a male, this is a female. Males and the female gets pregnant, and you can see patches. This red patches shows you that these are eggs that the female is wanting to release into the environment again. And the cycle goes on and on. This is the life cycle of Pratylecus penetrans, pretty much the same. I won't bother myself here so that we don't lose time again. So you find here the different effect that this root damage can have on different crops. This shows you the effect of Belolimus longicordatus on the yield of cotton. You can see that as the population of the nematode increases, the yield also declines. This one is on tobacco. So yield loss as a result of Globodera tabaci. So the Globodera tabaci is also the sedentary endoparasitic. Is also a sedentary endoparasitic nematodes. So you find on this y-axis the weight of the shoots, as you can see. Then here you have different population of the nematode. So as the population of the nematode increases, what you find is there is a decline in the shoot weight. Right. Okay. So how do nematodes damage plants? Through direct feeding on the plant roots, right? Through malformations they cause on the host tissue. They also can predispose this host plant to other physical stress. Imagine wounds on your body. What happens is just anything can penetrate if they are not cleaned properly. So with these damages on the roots or even on the leaves, as you find for Aphelincoides, this can predispose the host plants to other physical stress. It can even serve as a basis for entry of other secondary pathogens. We call this disease complex. And it could lead to breakdown of the resistance of the plants, and then makes it susceptible to other pathogens. What about the issue of vectoring? You can find that vectors can have access as a result of this nematode. There are some nematodes that carry viruses. Right. There are some nematodes that carry bacteria. So, and that is the problem. So they transport these things. When they feed on the plant, they also introduce the virus they carry in their guts into the plants. They can also lead to the suppression of beneficial organisms, depending on their population. So if you have other beneficial organisms in the soil, but the population of these plant parasitic ones are higher, it could be a problem, right? So again, I don't think I should bother you so much. I will try to share this material with you, but I thought to share my thoughts on nematode ecology and control. It's quite an interesting one. Of course, it depends on how much time you give to it. People have said nematology is difficult. Whoa. It has always been a case of people procrastinating. They don't want to settle down to study. For those of you working on environmental nematodes, I hope you are getting yourself conversant with these nematodes in the environment. For those of you working on plant parasitic nematodes, I hope you're also getting yourself familiar with the plant parasitic nematode and their ecology. So this is what I thought I should share with you. It's a form of introduction. In our next class, we'll take on some other specific areas. We have just 4 minutes left. This class was supposed to be a 45 minutes class, but I think we had some technical issues here and there. Okay,
Sir,
are there any general questions? Yeah.
Sir, please, I don't know. I am, my program is plant pathology, and actually I don't know if this CEP 825 is for me. Like, I don't know, because I checked my timetable and it's not there. And the course is more and about the plant pathology stuff.
Yes. So as I said, most likely, the reason I wanted you to take this course is because there are some things I will say in that course that have been captured here. So I don't have to repeat myself all over. You will also share the course synopsis with me. Please send me the course title and all that. And it's good you raised this. I've always told people, when you hear pathology, pathology doesn't refer to the study of only fungus. It's a misconception. Pathology has 4 various components. It has nematology, it has virology component, it has mycology component, and it also has bacteriology components. Okay, Matthew. So what you have been exposed to is the nematology component of that course. But it's good you raised it. We can also take your next whatever, we'll take that one up. This is an introduction where I want all of you to just join so that you can have a broad idea. By the next class, we're not going to be having a combined class anymore, because then everybody will break into their specific areas now. But for this one cuts across. Is a general introduction. In fact, if we know, we'll have opened it up. Though anybody that is interested in knowing what nematology is, they can join this class. It's just generally an introductory class. Right?
Thank you, sir. Thank you, sir.
Good one. All right.
Thank you very
So
much, sir.
I have 2 minutes left.
Sir, I don't know whether you'll be able to send us the records of this class.
Okay, I will try. I don't know where this thing is recording, but I will try to check. I don't know where it's recording, but I will check. I think it's recording on my system.
Yeah, I started
Yes,
recording it, but I didn't start from this.
Okay, are you the one recording, Matthew?
Yes, sir.
So please, you share with us. So.
But I didn't start from the beginning. That's what I'm saying.
2 of us cannot be recording now. It's only one person that can record in a Zoom meeting. Everybody cannot be doing recording. So I don't know
from where you started, it's okay.
All right then. Okay, so thank you.
Will the material be shared?
Of course.
Okay.
Yes.
Thank you.
All right. So thank you all. Have a wonderful evening.
Okay.
Yes, sir. God bless you.
Okay. Bye-bye.
Right.